Okay, hello everyone. I'm Seth. I'm a postdoc at Notre Dame in the Department of Physics and Astronomy. I do theoretical particle physics and cosmology, and I'm happy to welcome you all to the second edition of CHATS, Culture, Humanity, and the Arts Through Science, here at Culture Café. We'd like to thank the Notre Dame Department of Chemistry and Biochemistry for sponsoring the event. And I'd like to thank Stephen and Mary Merriman for inviting me to develop this series of events and for all of their support and organization and their keen interest in arts and science. So the event, as you know, is free, but a great way to support the Merrimans is by enjoying some of their fantastic goodies in Culture Café right outside. You can also, if you're so inclined, donate directly to Merriman's Playhouse, which is the nonprofit that owns this space that we're using. But without further ado, I'm happy to introduce our special guests for this edition of CHATS, Professor Saurja DasGupta and Argha Manna. Okay, so Professor DasGupta is a professor in the Department of Chemistry and Biochemistry with a concurrent appointment in the Department of Biological Sciences. He got his PhD in chemistry at the University of Chicago, working on RNA, and then did postdoctoral work at Mass General and Harvard Medical with the Nobel laureate Jack Szostak. Saurja joined Notre Dame in 2024, and I had the pleasure of meeting him about a year ago. Argha Manna has a Master's of Science from Calcutta University in Human Physiology, Microbiology, and Immunology. Was a senior research fellow in the Division of Molecular Medicine at the Bose Institute in India, where he worked on cancer biology, and has decided in recent years to devote himself to art and to communicating science through art. Argha has worked on many notable projects and has held appointments at the Indian Institute of Technology and at MIT here in the States. And he's currently here to collaborate with Professor DasGupta on illustrating the origins of life. So welcome to both of you. Thank you for joining me.
Thank you.
So to start, I think a good place to start is that really everyone should be amazed at the enormous amount we have learned in the past few decades about biochemistry and about how life works. And so for those of us, the lights, yeah.
Illuminate your
yes,
place.
Illuminate, very good. For those of us who have not taken a biology class since high school, can both of you talk a bit about the types of detailed knowledge we now have about biochemistry and life, and about what modern biochemistry research looks like?
Sure. Can you all hear me?
Yeah.
Good. Okay. So you're a physicist. Physics is an old science, right? Biology is actually a new science because, as far as I can tell, I'm not a biologist, by the way. I'm a chemist. I'm a trained chemist. But I think biology is fertile grounds for different fields to sort of come and contribute. So biology used to be originally a descriptive and classification-based science that people saw life around them, and then they grew that. Actually, art comes into play a lot here. So it was description, classification, and some art. But I think the molecular nature of biology started to emerge in the late 1900s and early 20th century, much like physics, started this new physics era in the early 20th century. Biology started becoming molecular, and this is a good question because I just taught metabolism to undergrads at Notre Dame this semester, and I was telling them that there was this guy called Ed Buckner who made had this experiment which got him the Nobel Prize, where he took yeast cells. So we knew at that time that yeast fermented sugars, right? But he took yeast cells and he ground them up, and then he added the sugar, and it still got fermented, which told him that you don't need life or living cells to do the chemical or perform the chemical process. You can just have components of life, right? So that was the birth, I think, of biochemistry, and that was a big step because that was a there was a strike to the vital force theory, that there was nothing special about biology. It was basically chemistry within, right? And then all through the next few decades, we had a discovery of enzymes, lots of metabolic pathways were discovered. So I think biology started to become more molecular and more chemical, right? So it was not descriptive anymore. And then, as many of you know, so there was this year, 1953, when something very, very important happened. If anyone care to say what or guess what happened in 1953? It was probably the most important discovery in biology ever made. Exactly, right? So we got the structure, the 3D structure of DNA or the 3D structure of our genes, right? So that kind of led to the formulation of what we know as the central dogma of life, which is basically the blueprint that all life uses, all existing life uses the same blueprint, molecular blueprint of life. And that was in the 1950s and 60s. We figured out the genetic code, which is basically, again, the computer code that runs in all of us. That was a big, big discovery. And then once we have the molecular blueprint of life, once we have the genetic code of life, we can now think of actually interfering with these biochemical pathways, right? Because we know these molecules and how they look, how they work. So now we can think of in an informed way, sort of interrupt these molecular pathways and for therapeutic purposes, for example. And then more recently, we have these things called gene therapies, where we can actually interfere with our genetic molecules, right? And we can actually edit out faulty genes even before they make proteins. The best example, obviously, is CRISPR, which is now gaining a lot of steam. So going from Carl Linnaeus drawing these things and classifying different types of life forms to actually editing our genes and actually having therapies based off of gene therapies, it's amazing. And it's all because biology became molecular. And as a chemist, this makes me really happy. This is something that actually annoys a lot of biologists when I go and tell them that biology is basically an elaborate contraption to make the chemistry happen. But I actually believe that.
Yeah, my opinion?
Yeah, please.
Yeah, like I showed you already, that's told you everything. So biology, yeah, of course it's about classification initially when it started and then moves towards molecular things. So as an artist, now I'm an artist. Of course, this when this thing happened in 1953, it was kind of a pivotal moment. You know, like when people started understanding about the molecular nature of life was the thing. And Saurja was talking about central dogma. So this is central dogma. If you can see this is, and so this is the central dogma, like kind of a linear thing. Though it's not linear. So all these things are molecules. What is happening every time within our body? Molecules are interacting to each other and coding for our life. That's how cumulatively our body works. And because within individuals, some of these things are happening every time. And yeah, so actually you can see it of course it happened in 1953. Watson and Crick, they could decipher the 3D structure of the DNA through crystallography technique. So I would also say, like, since the development of physics happened before Second World War, so all those like outcome of the physics research, it was utilized in biology when the Second World War was done. So after Second World War, it was the time for biology, specifically molecular biology, and before that it was time for physics.
Right. So you mean like in having learned how to understand crystal structures?
Exactly. So everything, like all those kind of techniques, all those like understanding about atoms, the scattering, all those things, light, how the molecular light play together. And it did not stop there. Actually, they really worked hard to understand. Of course, the one moment when the one structure is discovered, it cannot be the end, right? They have to understand the entire story, and it happened in 50s, 60s in this way.
So can you talk us through this central dogma? We have these three different kinds of molecules.
Yeah, sure. It's the Father, the Son, and the Holy Spirit of molecular biology. And you'll see that it comes down to one, just like Holy Trinity. But the thing is, before that, actually, I'd like to mention, since you're a physicist, you might be happy that after all the great quantum revolution happened, and these physicists were all euphoric that they made some huge contributions, then they started to explore the new frontier, because these are curious people that just constantly want to generate new knowledge. So people like Schrodinger actually wrote the first book on, so his book was called What is Life? So he was trying to figure out what life was in a scientific manner for the first time.
Which is still a great read.
It's a fantastic
highly recommend
read, yes. But he didn't understand much chemistry, but he was hitting the right points. And then people like, for example, George Gamow, who's a black hole, right? A black hole?
Yeah, cosmology.
Yeah. So he was one of the main sort of players in discovering molecular biology or the new biology. So yes, physics had Francis Crick was a physicist, right, who discovered one of the discoverers of the structure of DNA. But okay, to come to the central dogma, so basically what happens is that all of life that we have so far has this this blueprint, this molecular blueprint that's running inside of our cells, where we have genes, right? Genes tell our cells what to do, what stuff to make, and how to function. And genes are usually made of a substance called DNA, which we all know. You know, music is in our DNA, it's kind of in the common parlance. And then the trick here is that the genetic information that's written within DNA, the cell cannot read, which is weird, right? Why would the cell code genetic information in the DNA when it cannot really read that? So what the cell does is it rewrites that genetic information in the language of another molecule called RNA, which was in all of your COVID vaccines. So it looks pretty similar, but slightly different. But RNA is basically what my lab at Notre Dame studies. And then the cell can read the RNA message and then use that message to make the proper protein molecules. And the proteins are basically the workhorses of the cell. They would catalyze chemical reactions, so that help you digest, talk, breathe. Everything you do, proteins are important for that. You know, they're also in your hair, skin, muscles. So they also build things. So this is the workflow, right? So DNA to RNA to proteins. And the interesting thing is that this is happening, the central dogma is happening in bacteria and also in blue whales and every form of life there is. So basically this is what I tell my students is, you know, all life runs the same operating system, but in different form factors. So we are basically the same thing that's happening in each of our cells. So this is what was, so this was if I'm not mistaken, this was 1957 when Francis Crick. 56, okay, good. So plus minus error bars. This was a lecture he was giving, I think in England somewhere, Cambridge probably. And he was talking about how the genetic information flows. And at that point we just knew that DNA had that genetic information and RNA had something to do with the flow of genetic information. So what he was saying was that you can have DNA information go to RNA and then RNA information go to protein. And then DNA and RNA are a class of molecules called nucleic acids, right? So they have this thing called digital base pairing, which is again something that I don't want to go into in details, but these are called nucleic acids. Proteins are a different kind of molecule, right? So what he was saying was that information can go from one nucleic acid to another nucleic acid. So DNA to DNA, which we know as replication, DNA to RNA, and then RNA to RNA, and then RNA to protein, never from DNA to protein, and never from protein back to RNA to DNA. And you have to understand that we hardly knew anything about these molecules when he was doing this. This was purely based on intuition, and Crick was one of the best intuitors of all times, I think. And this has now become, and he actually, well, the central dogma was a sort of a snide remark toward the Catholic Church. When he made that, he was actually making fun of them. This is why he called this the central dogma.
Let's move on from that. And so on the same theme, you've already brought up RNA and the curious role it plays sort of in between DNA and proteins here. And I understand your lab is focused on understanding RNA and in particular the connection to the origins of life. So maybe we could talk. You could talk a bit about what we know about how life began, or what we strongly suspect but still needs to be confirmed. And this theory I understand you work on that it was RNA that came first before DNA.
Yeah, that's a whole lecture, multiple lectures,
Yes.
maybe a course someday. Well, it's also there. So life exists. That's the fundamental place where we start from. So life exists. So however people say that, you know, the origins of life is not a very likely event. The probability is very, very low, but it has still happened. Like, what was the probability that all of us would be in the same room at the same point? Very low, but it happened, right? So things happen. So starting from there, there are many sort of models to explain the origins of life, and I have to say that we don't know for sure what really happened. The one reason being we don't have fossils of the earliest forms of life, right? So that's a problem. So we have fossils of, say, dinosaurs obviously lived and died 65 million years ago, but life started sometime around 4 billion years ago, which is a longer period of time. So it's deep into the past, right? But the thing is that we do see life around, just like physicists study the cosmic microwave background radiation as an evidence for Big Bang. We have much more. We have life that exists. So we can study them at the molecular level and make intelligent guesses. So I can tell you what my favorite, and I think this is widely accepted model of how life emerged is. So we think that life, so how long do you have for me to answer this question? 3 minutes.
Sure,
3 minutes.
please.
Okay.
Go ahead.
This is capturing 4 billion years of existence. But the thing is, so when the Earth was formed, that was 4.6 billion years ago. It was really hot and warm and volcanic eruption. The crust was not properly formed. Lots of heavy bombardment from space, right? It was not habitable for life at all. But things started to cool down, oceans started to form, temperatures and weather started to... Oh, do you have an image for that? Oh, there you go. Fantastic. That's the benefit of having an artist around.
Yeah.
So this was an Earth where life probably could be formed. And then we have the first evidence of, the earliest evidence of life is around 3.5 billion years ago. These are not direct fossils, but called microfossils in rocks called zircons. Maybe lots of these rocks are found in Australia. So we think that, so that kind of life, the life that we could detect, also use the same metabolic pathways as we do today, which means that they also use the central dogma that I just talked about, DNA to RNA to protein. But proteins are notoriously difficult to make. So the earliest forms of life, when it first emerged, would not have used proteins, right? So we think that the earliest forms of life were very simple, much more simpler than what we have now, even much simpler than like a single bacterial cell. So we think that the atmosphere of the Earth around 4 billion years ago had lots of ammonia, methane, hydrogen cyanide, carbon oxide, so carbon dioxide and carbon monoxide. And we have shown that if you could mix them in the right manner, then you can get these things called nucleotides, which are the building blocks of RNA. So if you have a necklace of RNA, then these are the beads. And then you can actually these things can self-assemble into short pieces of RNA, and these short pieces of RNA can link up together to form longer strands of RNA. And what happens in biology, these biomolecules, these biopolymers, is if you create a biopolymer that's long enough, they will fold on itself. This is just by physical forces. They would crumble up and fold into these globular structures. And whenever biomolecules form these structures, these complex structures, some of them are bound to show some function, right? So for example, they can start behaving as enzymes. So we think that the first enzymes were based on RNA. So these RNAs linked up, formed these globular structures, and you can imagine the vast sequence space of RNA, the combinatorial space of these molecules, and the time, like we're talking about millions of years here. So we could obviously generate these functional enzymatic RNA molecules, and then these RNAs would start to copy themselves in other molecules. So you'd have these molecules that would self-replicate, and then eventually they would be encapsulated by lipids. So lipids like fatty acids, like soaps, right? If you have soaps, you can make soap bubbles. So you can have fatty acid molecules, you can have these sort of bubbles called vesicles, and capture these RNA enzymes inside. And I'm going to take 30 more seconds. So what would happen is that you'd have these vesicles with these self-replicating RNA molecules. Now you can imagine if these RNAs start to generate more copies of itself, very soon the vesicle would grow fatter, and then ultimately split, right, into daughter vesicles. We call them protocells. And when these RNA enzymes copy themselves, they make typos, they make errors, right? And not all typos are bad. Some typos, some mutations actually make the enzyme more powerful. So you can very easily imagine in a scenario where these bubbles are growing and dividing with RNA enzymes inside of them, those bubbles with better enzymes will make more copies of themselves and may grow and divide faster, and then their lineage would very easily take over the entire population. And what this is basically natural selection, or basically evolution by natural selection. And when a chemical, this is all chemistry, right, so far, so when a chemical system starts to evolve in a Darwinian manner, we actually call it biology. So this is the point where we actually have biology from chemistry, and this is how the earliest forms of life would have emerged. And this is the dream experiment that we want to do in the lab, and I have some students here in the back. Hopefully they can make that happen.
Very cool. Right, yeah. Well, and yeah, and once you have some ability to self-replicate, and if it's an imperfect process, indeed, it's off to the races. It seems like then something
It's an
more.
eventuality.
Yes, yes. And so, Argha, I think you're illustrating here this theory that life began in tide pools.
Yes.
Right.
Yeah, like kind of like close to 4 million years ago. Like, how was the landscape? No, sorry. Yeah. Around 4 million years ago, how the Earth looked like. This is the landscape. So we really do not know about that. So it's all imagination or some kind of speculations. So this is kind of a volcanic island. It's emerging up from the sea, and all these things are kind of like hot water ponds. Charles Darwin actually predicted that. Actually, along with Darwin's warm little pond. And here you can see some meteorite is coming on Earth, and some little water bodies. And all this that Saurja was talking about the vesicles. These are the vesicles in water. And actually, what happens from the recent research, we can predict that. We cannot really know. There are some kind of a dry wet cycle where vesicles were formed, and then again they transform into a gel-like layer. Again, they transform into a vesicle when the waters come. So it's kind of a cycle. In this way, vesicles survive, and within the vesicles, RNA also survive. That was very much important to survive within that, because RNA, molecule like RNA, like within that kind of a hostile environment, if they didn't survive, there wouldn't be any life.
I think there's some irrelevance to these things being small, because you can imagine ultimately things need to come together to create new things. And if it's an ocean, like this is a big debate. Like, where did that life start? Like, is it in the ocean or lake or ponds or hydrothermal vents or wherever on minerals? So if it's an ocean, then everything is so dilute that it's very unlikely that these molecules encounter each other and form new products, right? So I think these small water bodies are really, really good because there are these temperature gradients, wet-dry cycling, which is important for forming large polymers. So these are all not just, I would actually say this is not just imagination, but these are from scientific research.
Yes, of course. Little bit imagination, little bit data. Yeah. So simple, like small test tube and lots of molecules to interact. A large pool and small number of molecules, it didn't happen.
Right. Yeah, so last year I read a very interesting book by Nick Lane. And he argued for a beginning of life in ocean vents and in the physical structures around ocean vents. So not just some random place in the ocean. Is that, you think that's definitely not true?
So this is the vital question?
Yes.
Yeah. I like the book. He's a good. So his argument is there's these two camps. This is technical, but some people think that all these metabolic pathways that we have started initially, and then the genetics started, and then we had life. So I don't know how you could call a metabolic cycle life. As long as if you don't replicate, I don't think you're life. If you don't evolve, I don't think you're life. But it's kind of cool to think of metabolic pathways without enzymes, without living cells. So that's Nick Lane's whole thing. In terms of hydrothermal vents, again, his idea is that if you have a hydrothermal, by the way, those of you who don't know, you have pictures of hydrothermal vents? You should. So these are very nice structures under the ocean where you have
Warm little pond.
Yes, but we have these gradients. So you have different acidic and alkaline environments close to each other. You have hot and cold environments close to each other. So life needs gradients. Life needs to go out of equilibrium. These are technical things. He thinks that those are important. But I think if you consider life to be something that replicates, right? So you would need some sort of polymer, some sort of genetic polymer, some sort of cell-like structure. None of this would survive in the temperatures that hydrothermal vents provide. So I don't think life would emerge in hydrothermal vent scenarios, but you could have metabolic pathways that eventually could have led to the first cells, but not life. It's impossible if that's a short answer.
I see. Okay, interesting. I'll have to think more about that. So, Argha, I wanted to ask very generally, you started as a researcher in cancer biology, well, you started before then, but you were a researcher in cancer biology,
Right.
and you've made this transition into sort of full-time art, but still with science and explaining science through art. So I wanted to ask how that happened and why.
Okay. Well, so during my research day, my main instrument was laser scanning confocal microscope. So I was very much fond of image, So I was a little bit bored about like all the gel data, like black bands on some paper sheet,
Yes.
or film. So at the time I didn't know that, like image would be my career,
Sure.
dealing with image and not capturing, the drawing. What happened in 2015, it was the 100 years of general theory of relativity, right?
Mhm.
Right?
Yes.
So Science magazine published a comics about general theory of relativity. Okay, comics, it was a review article, but made in comics. And the same year, Nature published an article on climate change. And so I was surprised, in Nature and Science, they were publishing comics. Okay, so already I was not very much liking the benchwork. I wanted to be a little bit creative. And this was a very pivotal moment. And who created the article, published in Nature, is Nick Susanis. Now Nick is a very good friend of mine. And Nick, the next year he published the first PhD thesis in comics from Columbia University.
Wow.
And Harvard University Press published, in their entire history of publication, first comics book. It's called Unflattening. And it was based on mathematics, It was the antithesis of Flatland. So these things, affected me a lot. And then I thought, okay, comics would be my life. But since I was trained in science would be my subject, but my dissemination process would be comics. That's the thing.
Very interesting. Last month I was speaking to another physicist, and we talked a bunch about how our experience of physics research, in fact, includes a lot of creativity, we felt. You've just alluded to your research maybe not having anywhere near as much a creative element as you have now. Did you, or do you see some creativity in the research that you have done or do you think about it differently than physicists do?
I think at the highest level, science needs to be creative, otherwise you're not going to push boundaries. But I honestly think, and I'll be very frank, I think, so my area of research, which is biochemistry, I don't think needs that level of creativity as, say, a theoretical physicist needs to have. We are more about getting our hands dirty and trying different things and seeing what works. Because if you do experimental science, especially biochemistry or anything to do with biology, you'll realize that biology is extremely messy. Cells don't really follow neat rules like even cosmic or planetary bodies do. So we have to get our hands dirty and do things, and oftentimes what you think is a clean, neat idea, like the elegance of an equation, for example, doesn't work in biology, which is why a lot of these physicists in 1950s and 60s came up with these really neat models for the genetic code that had to work in their mind because the science was so elegant, but turned out to be all wrong.
I think in biology also there is room for creativity. Of course you can predict something, and of course cell is messy. That's why cell is very abstract. What is happening inside cell, But yes, I agree in one point. Theoretical physics is dealing with a little bit more abstraction, like pure mathematics is close to that, and biology, of course, you have to prove it. But again, what is happening inside cell, we really do not know exactly how it's going on.
Sure.
Every day we are discovering a little bit more organelles, we didn't know that. Things will be changed. But how cell is dealing with their surrounding environment, that's imagining that things should be very creative. Then actually you can prove it.
And so on art, at some level, art is everywhere in communication between scientists. I mean, I've had lots of meetings where mostly what we did was draw pictures on a board and try to explain to other people what our pictures represented. Is that style of communication also very common in biology, biochemistry, and how is artistic communication about science for other scientists different from communicating it to the public?
Okay, so of course, let me share one image. First of all, anyone can identify who is the artist? Yes. So what Da Vinci was trying to do, and of course, at that time, in 15th century, there was no such term as scientist, right? All of the philosophers. Even that term perhaps came later. Yeah, you know. So what Da Vinci was trying to do was, he was, at least for this series of drawing, where he did a series of fluid study, any kind of fluid, water, cloud, air, any. So of course it was commissioned by the king because they wanted to make some dam on some river. But what Da Vinci was trying to do was trying to find out patterns, because like the mathematicians always do that. So, but now we are considering this kind of a communicative piece. If someone really wants to study some abstract like fluid dynamics of a fluid or Newtonian, non-Newtonian, whatever, they can use this, get some reference to that. So it's for the scientist or the public, it doesn't matter. You know? But for scientists specifically, let me show you one thing. Like this one. This one of the part of my project at MIT. So what we are trying to do, we are trying to make a new document for WHO for future pandemics. Touch it, we really don't want that, but if it happens, how actually disease transmit. Okay? So all the WHO's, all the posters they make, the CDC's posters, all was wrong because it was not scientifically correct. But how to deal with that art so that public can understand it. And not public, scientists can understand it, what is going on. You know, so I worked with Professor Lidia Buriva at Fluid Dynamics of Disease Transmission Lab. She's a mathematician, mathematical physicist, and I was an artist. So we created all those images. These are all hand-drawn but based on data. You know, how within a room actually disease transmit. Particularly for this one. This image is very much important. It's not that in air, aerosols loaded all the virus or vectors are floating. No, within even air, they create little bit pockets. And as a little bit pocket cloud, it transmits. So these things are for scientists, because common people really do not have to do anything with that. Of course, it's perhaps a little bit interesting drawing like Kusuma's dot artworks. If you know about that famous Japanese artist. When I drew that, I was inspired from Kusuma. Like I was thinking like how from that heavily loaded data, lots of millions of data points, I can create something so that scientists and public both can understand it. That this is how. So I took direct reference from her. You know how art inspires. But this is for scientists.
But this is from high-speed camera data, right?
Exactly.
Right.
Exactly.
So this has scientific.
Doctor is scientific.
Yeah.
Actually, it's aerosol transfer. Now this is another one for scientists. You know, these are the best molecular artist I would say ever born. His name is Arvind Grace. You know, now if you go to Harvard Medical Institute's website, they have all his artworks. He, at that time, of course, there was no computer. All these are oil paints or some acrylic or some watercolors. He drew all the important molecules. This one is cytochrome C, this one is one pocket of myoglobin. And in 60s and 70s, American textbook, all was filled with his artwork. At that time, there was no computer, so we couldn't create some simple diagrams. These are for public, but even scientists were benefited from that, you know, the cytochrome C. So this is the thing. So, like for public or for scientists, there is some balance. Some are a little bit more scientific, some are a little bit more communicative.
And so, whenever you're communicating art, there's some need to simplify, to leave out details. How do you think about what things you focus on and what things you discard, or do you worry about discarding too much?
Yeah, so there's a long debate actually on that. You know, like it should be diagrammatic or naturalistic. The debate is coming from during the time of Albert Dürer. Like there's a famous painting by Dürer, it's on a grassland. If you just Google it, you can get it. It's so detailed. So everything about a grassland is there. But is it scientifically communicable? Like, can a student can understand or extract the information from it? So there is a debate. I don't think like diagrammatic artworks, which usually we can see in textbook, is better, or the actual naturalistic artwork where we are not missing any information. There is a debate. So you have to understand who is your audience and for whom you are creating and what is your purpose. That's the thing. Again, so there is no such straight answer. Which one is better?
Sure. Custom for every
Yes,
situation.
yes.
Interesting. Now, I want to return back more to the origins of life. As you were saying, it seems life began very soon after the Earth cooled down enough that you could stably have any sort of molecules. What do you think this means for the abundance of life in the universe? And do you think there might be many basic mechanisms for self-replication, and the first one that formed here is the one that everyone uses, or do you think that we landed on the only thing that's possible?
So two questions, right? The first one is, are we alone in the universe? Which is the big question, and you're better to answer this than me because you know the number of galaxies and the number. So I think there are like 100 billion galaxies, right?
100 billion stars.
Right. So you can do the. So I think 1 septillion planets or some. No.
Something
like
that.
that. 1 to 24.
Yeah.
Right. Some.
Yeah.
So it's highly likely that so let's say if 1 in 1,000 or 1 in 10,000 planets are Earth-like, right? Then you still have so many planets that you could have life in. So statistically speaking, I think there could be life in the universe, and there could even be life just outside of our solar system. But we can never break Einstein's speed of light, so we can never really go there and find out what happened. That's a problem, right? So yeah, I think that there should be life on other planets somewhere in the universe. And the second question is, would that life use the same chemistry as ours, right? So again, there is no need to think that it would use the same chemistry. Like, so for example, we have nucleic acids that form genes, right? They can replicate. But you can have, and in fact, chemists are great at tweaking biology, right? So RNA and DNA look a certain way. There are 4 kinds of beads, building blocks that DNA and RNA are made of. But chemists can synthesize many more analogs, we call it, right? So these are kind of like what exists, but not really. And they've shown that you can actually make nucleic acids or polymers based off of those building blocks. And actually there was a study in 2019 that I was part of that created a lot of noise, where so DNA and RNA are made of 4 building blocks. We call them ATGC and AUGC. But we created a DNA and RNA made of 8 letters, right? So we expanded the genetic alphabet from 4 to 8. Now that group that was leading this project has now made it to 12. And so you can have 4 natural and 8 unnatural. So you can create these kind of polymers using something else, and then you can imagine in a different planetary environment you could have different feedstock molecules, different gases, different building blocks, and that could create some sort of polymer which is different from what. But again, this is something I think I mentioned in the last CHATS session, is that I think whatever the chemistry might be, it would still evolve in a Darwinian fashion, that is by natural selection. So replication, mistakes get selected and then propagate. So I think that, there are no laws of biology like there are laws of physics, but if there is any law of biology, then that's Darwinian evolution. That's as close as you can get.
Very interesting. Yeah, I'm inclined to agree. Of course, what we find on Earth is so particular to the history of this region of space and what the average star was like that went supernova and caused the nebula that we formed from. So indeed, there should be some statistical variation, some variety of things in different regions.
And I would like to believe there's one beast, right?
Yeah.
There's one planet out of all of that could have life.
Yes, of course, or even moons. Yes.
Definitely moons. Yes, exactly. So Europa and Enceladus are 2 moons that Jupiter's and Saturn's moons that scientists think you could find life in, but I'm not that optimistic. But NASA is carrying out missions
We do have evidence for liquid water, though.
Sure, yeah.
Yes, yes, yes.
That's not quite enough, but
Sure, of course, of course.
yeah, starting point for sure.
Of course. Interesting. Yeah, that research sounds very cool. I'll have to go back and read your paper.
Sure.
Very cool. So I have a question, maybe a little bit out of left field, but so every so often in the United States, there's an opinion poll conducted by a firm called Gallup. And they ask every so often about beliefs in human origins. They're asking the population of American adults. Do you know, or would you like to guess, what percentage of US adults believe that humans were created in their present form less than 10,000 years ago?
About, I believe, 40%?
Indeed. In fact, when they did this poll last year, it dropped to its lowest level ever of 37%. So we're making some inroads. And indeed, but this point of view is very common. I was raised with this sort of belief system. Have you encountered, both of you, people like this, either in the U.S. or India or elsewhere? And what do you say to such a person?
So that's interesting because again I grew up in India and the whole maybe I call it cult, but category of people that are evolutionary deniers is non-existent in India in that form because there is no Bible that has a chapter called Genesis which can be literally interpreted in Indian. So India is mainly Hindus and Muslims and some other religions. But there is really no religious text in Hinduism, no one religious text in Hinduism. I think in Islam there are certain things that are similar to the Bible's Genesis, and I have encountered people not in India actually, but in the U.S. So I was in Chicago for my grad school, and I was part of the Spiritual Life Council at the University of Chicago, and I spoke to people of different faiths, and I had debates with a student there that refused to believe in evolution. And the reason basically that was given to me was that it was in the text, right? Because there's so much evidence that the Earth is now. I mean, the oldest rocks are 3.5 billion years old. Yes, but I think this is more in the US than in India, which is kind of maybe counterintuitive to you, but yeah.
Well, I think in the last 15 years in India, evolution denial has been increasing. Yeah, because of course, actually in Hinduism there is no religious text, but like in last 15 years, people started considering all those mythological texts as their religious text. So
BGP stuff.
yeah, right. So there are many evolution denial. But what is their counterpoint that we really need to not know? They just deny something like God created us. The Hindu gods just created us as we are. Also in India is suffering from caste system, so they got kind of a alibi to support that. God created all this group separate and this group separate.
Although actually I should mention that when I was a PhD student in Chicago, there was you must have heard of, there's a group called ISKCON. I think in America you call them Hare Krishnas. Right, so they have scientists, PhD scientists working with them, and they try to publish, let's say, in peer-reviewed scientific journals. It's basically science articles with science terms, but at the end would end with so there are so many faults. It's the God of the gaps type of argument. So the Indian scripture actually, if you study that, you would actually end up with something like intelligent, not creationism, but something like intelligent design, which is kind of the same. And that a couple of those papers were actually published, and that was 2015, I believe. And I was reading Christopher Hitchens and Richard Dawkins and Sam Harris and Jerry Coyne and Dan Dennett. So I was very quick to get angry at paper. And I spent an entire night in the library and wrote a rebuttal to that paper and got that published in the same journal. And which for that, I had to actually ask myself, do I really know what evolution is? Because I would wager that a lot of people that think they believe in evolution don't quite understand evolution. That's also kind of a faith kind. Scientists tell us evolution is real. that's why evolution is real. So I had to really challenge myself, and I was a chemistry PhD student, so I knew a little bit about evolution, but I had to really ask myself what the fundamental features of evolution was, and then I had to write that article, which kind of strengthened my base and knowledge base of what evolution was really about. And then my lab now studies test tube evolution, so I think that was important.
And of course, it's not only these religious texts. There's this intuition that many people have from sort of a broader cultural milieu that, you know, living things, you look at them and they're just so obviously different from non-living things. How it seems impossible that you could have gone from one to the other. And people, you know, these days people make very similar arguments about consciousness as well. So, well, maybe even more broadly than that point, you both have collectively many years of education and research in biology and chemistry and biochemistry. How has this affected your own thinking about life or about humanity, yourself?
Okay, so of course studying biology, biochemistry, molecular biology, I think made me better human. Why I can explain, like about skin color. So when I knew about the proper scientific explanation, of course I was immune to any kind of bullshit idea like two people are different who have different skin color. No, they are not different. They are perhaps what kind of biology is going on at the skin, at molecular level is different, and it's not a big deal. No, but we as a human we are not different. Or like how I deal with women. You know, so of course when I was in school, I studied in a boys' school. Like women are different, we are superior, and studying biology, of course, their systems are different too. It's had to be, you know, but we are same human. So that's how I think studying science, studying molecular biology, it's made me better human. That's what I can say personally.
I think that from an even deeper point of view, because I was talking about life's origins. So if you, like I said, the same blueprint in all of us, right? And if you use that information, that the fact that we have the same blueprint actually indicates that there is, if you have tree of life, it has the same root, and we have a name for the root. We call it Luca, or last universal common ancestor. So we actually emerged from a single population of Luca, which, I mean, if that is not the most connecting and unifying thing, that we all came from a single kind of life, and we have the same root, we have the same chemistry, the same molecular biology in all of us. I really don't know what could be a more unifying.
Right. There are animals also.
Yes.
All the species, like, you know.
Yes.
So we are not the superior species.
Yeah. You know, and I think very much to both of your points, something that I think is very damaging about this view that the universe and humans were created only a few thousand years ago, is that then you have this worldview where for the entire length of the universe, different humans have just been different, right? Humanity split apart at the beginning of the universe, right? Whereas, as you say, the actual comparison is that life has been going on for more than 4 billion years on Earth, whereas we are related to every other human in the last 200,000 years or so. Indeed, I feel for myself as well, this understanding has helped me connect with humans across a broader spectrum than I would have thought of doing as a child.
And evolution is a huge consciousness raiser, right? So just like, you know, we thought that, you know, Earth was the center of everything, and then that was proven wrong. Then humans, it took us 99.9% of all species that came on the Earth and walked the Earth are now extinct, right? So this is something a lot of people that I've met, some more educated, scientifically minded people have told me that, you know, this is the purpose. This was the purpose, to get to humans. It didn't really, it wasn't really created out of mud all of a sudden, but this was the mechanism by which the goal was to create humans. And I tell them that if this was the goal, then this is a pretty bad mechanism because you killed off 99.9% of everything that ever lived. That's such a waste. So this is my view. But, you know.
Also, in general, like I'd take the opportunity to show you some image. Yeah, so studying science, like not for me personally, historically, how it could change the like human race or the understanding. Like this cartoon was published in 19th century in Punch magazine, London, when London was totally like smitten by cholera, you know. But at the time there was no idea about what make us sick. So they published that. It's all monstrous because that kind of myth was there. Then these are of course is illustrated by me, but inspired from the original sketches by Robert Koch. You know, so when he was studying cholera, you know, so slowly studying biology and understanding the living system, human race was coming out of some kind of myth, and this was how antibiotic was discovered, and this is how penicillin was discovered. It's a very important thing that happened, right? It saved life. So it's not only about philosophically what we think about other human or other species, it saved human race also, you know, from disease. So that I think we should mention. Of course, studying biology and molecular biology, it saved us.
Of course. Right, so one more thing before then we'll take some audience questions. As has been on display, you're together planning on or in the process of illustrating the origins of life in some comic or graphic novel sort of form. What do you both want people to take away from your finished product?
Yeah, that's in the future, but we're off to a good start, it seems. So there is no real scientifically accurate and scientifically updated comic, graphic novel that does this, that talks about the origins of life. So first of all, our big goal was we were very ambitious. Like we wanted to create an exhaustive, scientifically accurate and up-to-date comic book illustrating the origins of life that would use actual research papers and interviews with the actual scientists that did the job. Not speak in vague terms, not just one little pond and then the Miller-Urey experiment and then boom, creepy crawly life on Earth. That was not our goal because that's there in different blogs online. So we wanted to create a scientific document, right? That was in the main goal. And then the other thing was we wanted to, and we have these scientists that made these big discoveries as characters in that. So the way we have planned this is a Socratic dialogue where there's an artist and there's a scientist, and they're having this conversation. The artist is actually asking these pointed questions, and the scientist is trying to understand, answer them, and then all these historical scientists are coming in as characters, and we are seeing them perform experiments and making these discoveries, because we wanted to show, and this is mostly for students, I guess, and also for the public, is science is not this monolith that just happens. It's a living thing that really real people went through this process. These big giants in molecular biology make horrendous mistakes. They were just basically guessing and making intuitions, and some of them worked, some of them didn't. So what we're trying to say is that look at all of these things in its holistic way, and then see, try to extract useful information. So in terms of what I think we want the readers to take from it is something that I want all of you to take from it, is that the origins of life is actually a scientific problem, right? It has long been studied by philosophers and theologians. It is a scientific problem, and scientists are working on it, and we are actually much closer in creating life in the lab than you might think. So this is something I want all of you and the readers to know. And the second thing that I want them to know is that, again, science is a human endeavor. These are humans that are doing this. Science is almost never settled, right? No one is the science. Science is never settled, so there's always room for questions. And the third thing is that the whole scientific enterprise is extremely entertaining. It reads like a thriller novel, and it's a lot of fun.
Yeah, the same. Yeah, I agree with you. So as Saurja was mentioning, it's how we are conceptualizing our graphic novel is kind of a dialogue. So here is one sample page. Of course, for obvious reason, I haven't added text here. So you can see this is Saurja, this is me. We are having conversation. Text will be added. and we are discussing about different places. So in this way, the entire book will be designed like we are questioning each other and we are studying together. And another thing I want to add, like I am personally very much romantic about science, not only about data. So like a university building inspires me, people inspires me, old stories, artifacts. I'm very much it's very much important to me. Like I can show you just one thing, like this one, this page. So all these are from RNA type club boys. So he was talking about George Gamow. So, Gamo's image is not here, but this building, what they're thinking, papers,
this is Alex
books,
Rich thinking at MIT.
Thinking about MIT, about all those different possible conformation of the RNA. Like it's kind of a folded thing. So all these things, I'm very much romantic about the history of science. So that is also, I am expecting from our book that romance somehow will be conveyed to the readers.
Yes.
And if they get inspired, some kid can pick up science as their career. And being a scientist or artist is a beautiful thing.
Yeah. Yes, very well put. That sounds fantastic, and I'm looking forward to reading what you guys produce. Okay, are there any questions from the audience? Yes, Max.
This may be a better question for a geologist, but I was wondering if the studies of the origins of life considers if there was a difference in the elemental composition of the ocean in Earth's early years, or if there is evidence supporting this in either a way that could have facilitated the growth of life.
So the question was that I think that was the chemical composition of oceans in early Earth different from what they are now, and does the field take that into consideration? Right, is your question? Yeah, that is a better question for a geologist. Although I do talk to geochemists, earlier geochemists, so there was no molecular oxygen when life emerged, right? So because unless green plants emerged and started doing photosynthesis, we didn't have molecular oxygen in the atmosphere. So once we start to have molecular oxygen, then I would assume that the ocean had dissolved oxygen, which would definitely change the ecosystem of ocean. Other than that, I'm sure there were different minerals that would have been abundant in different times of the Earth. Also, I think once you have oxygen in the atmosphere, a lot of these metals, I mean this is again technical, but they exist in different oxidation states. So once you have oxygen, they start to get oxidized. Iron is a good example, and then you get a slightly different kind of metal, and that different kind of metal can no longer facilitate a chemical reaction that it would have otherwise facilitated. So that maybe half answers your question.
Also, another thing is when you have molecular oxygen, there is a high possibility to have reactive oxygen species. That for initial cells, when the life is at the nascent stage, is not very good thing. So I guess the first cell emerged a little bit earlier than molecular oxygen.
Much earlier. So
much earlier. Yes,
we think molecular oxygen emerged around 2.5 million years ago.
Yes,
so yeah, much
that's
earlier.
Yeah. Further questions?
Do you have a favorite definition or answer to the question, what is life?
I do.
The question, what is life? Yes.
Yeah, I do. So this is a NASA working definition of life, which was coined by a guy called Jerry Joyce, which is life is a self-sustaining chemical system capable of Darwinian evolution. So it has to be chemical, right? It has to sustain itself. Those are obvious, but it also has to evolve in a Darwinian manner. It cannot just replicate. So I give you a quick example. If you grow crystals of salt in a glass, or snow for example, if you have one seed crystal, it can form many copied seed crystals, but those crystals would be the same morphology as the same seed. But when life propagates, it generates slight variations, and that's how Darwinian evolution can happen.
Thank you. So if life is started in small ponds, was it only in one pond, or it could have happened in many small ponds?
So I think your answer is, did life emerge in just one pond or many? Yeah, definitely not one pond. But we're talking about a similar kind of pond. But yeah, There are many ponds. If the conditions are right, you'd have many. And actually, there is no reason to believe that the life that we are descended from was the life that emerged at the beginning. You could have a form of life that emerged and completely went extinct, and another form of life comes something like the universe, like, the Big Bang. You could have many kind of universes that I've read somewhere that those universes were not tenable. Maybe I'm out of my.
It's Penrose physics.
Right, but so there could be many forms of life that could have just died out, and the one that survived is us.
So that sounds like if you indeed create life in lab, how do you know that's how life started, our life started?
See, that's a great question. So the question is, if you manage to create life in the lab, then how can you say that is the way it happened? And the answer is we cannot, right? How can we? But what we are doing is, so the way I envision is that I talk to geophysicists and chemists, organic chemists, prebiotic chemists, and I want to get the inventory of all the stuff that would have been present 4 billion years ago, and I want to create life just using those molecules. So that's how I'm limiting myself. And even if you can create life in a way that makes chemical and biochemical sense using the stuff that would have been present in nature's kitchen 4 billion years ago, then at least you can say this is how nature could have made life. And this is like recreating the crime scene. Like, if you don't know how a murder happens, you still can reconstruct the crime scene, and that's our approach.
Great. Thank you, Saurja. Thank you, Argha. This has been fantastic.
Thank you, Simon. Thank you, everyone.
Fantastic. Thank you. Thank you, Steve. We've been trying to do this for a long time.
Yes. I'm very glad it worked out. There you are. Great.